Okay, welcome to another lecture. This one is section 2.3. We're going to be focusing on macromolecules. This one's a little bit longer, so if you need to pause and come back at another time, that would be fine. Okay, so organic compounds are anything that has a carbon chain to it. Carbon atoms that are combined with hydrogen, oxygen, and frequently it will also have nitrogen, sulfur, and phosphorus. So the main parts of an organic compound would be carbon, hydrogen, oxygen. Those are the key. Now there's always exceptions to the rule, and make sure you know these down here. So we have carbon dioxide, carbon monoxide, and carbonic acid. We saw that in our other lecture when we talked about pH. So those are not organic compounds. That will be a test question. Make sure you know that. And so they can be arranged. They could either be a single carbon, it can be a chain or a backbone, it can even form a ring, and we'll see that when we get to, for example, DNA. Okay. Macromolecules, macro means large, so it's a complex molecule with a backbone of carbon chains and then other elements are radiating off of it or other molecules or compounds. Okay, so there are four that we're going to focus on. We have carbohydrates, lipids, proteins, and nucleic acids, and I'm going to cover them in this order. So let's start with carbohydrates. Carbo stands for carbon, hydrate is water. It's also going to have hydrogen and oxygen. So there's that carbon, hydrogen, oxygen, okay, that makes it organic. All known types of living cells have carbohydrates. A carbohydrate is also a saccharide, okay, so you might have heard saccharides before. The building blocks, I'll be using that term, the building blocks of a carbohydrate are your simple sugars. Make sure you know the building blocks of each of these macromolecules, and I have a slide at the very end that kind of pulls it all together for you. Okay, so here are some saccharides. The first is monosaccharide, which means one sugar. This is your simple sugar. So carbohydrates, you can either put the building block as a simple sugar or they are monosaccharides. Most common monosaccharide is glucose, which is your blood sugar. And it's identified by how long the carbon chain is. So specifically for glucose, here's the formula or the molecule, structural molecule, carbon, which is C6, hydrogen H12, oxygen O6. C6H12O6. Now, it's identified by how long the carbon chain is. So it's 6 carbons, so it's identified as hex-OS. Hex is 6. Anytime you see OSE, that's referring to it as a sugar. Okay? Now you notice I have a 1-to-1 ratio. 1-to-1 ratio is referring to the subscripts of the number of atoms of each. So 6, 12, and 6, if I break that down, that is a 1-to-1 ratio. Okay? You will always find that in your carbohydrates, okay, or your sugars. So carbohydrates, depending on the molecule, could be a mono, a di, or a polysaccharide. So we've covered monosaccharide. Now let's look at disaccharide, which means two two monosaccharides put together, or a double sugar. So the first one is sucrose. Sucrose, you need to know that one. The common name would be your table sugar. It could also be cane or beet sugar. The two monosaccharides that make that up is glucose and fructose. So these two are monosaccharides. So the hardest part of this is keeping straight which one is mono and which one's di. You're just going to have to memorize these. Okay, maybe put them on flashcards would be a good idea. So I like coming up with little mnemonic devices. Let me pull up my little picture here. So good friends came over for sugar. So good is glucose, friends fructose. So one each, so those are monosaccharides, came over for sugar. And so you have lots of sugar, so that's sucrose or a disaccharide. Here I've got two different people, a boy and a girl, eating sugar or cotton candy. Okay, another disaccharide is lactose. I'm sure you've heard of lactose intolerant. These are people that cannot digest or break down the milk sugar. And so this lactose is made up of two monosaccharides, galactose and glucose. So a little mnemonic device, gals who eat glue laugh together. I don't know if you can see this image, but this is I love eating glue. I couldn't find two girls eating glue. I did find a couple of boys eating glue, go figure. But anyway, so the gals and the glue, those are two monosaccharides, and when you put them together, when they laugh, the two girls laugh, okay, that's going to make lactose. Okay, and the last disaccharide I'm going to have you know, there are a lot more, but I'm going to have you focus on three, is maltose. And this one would be 2-glucose. So see, it doesn't always have to be two different monosaccharides. It could be 2-glucose. And so malts are twice or 2 times as good. Okay, so maltose, 2-glucose. Okay, so that covered disaccharide. Now let's look at polysaccharide. Now this is a building of several glucose molecules together. Okay, so 3, 4, etc. And there are 3 main types: starch, cellulose, that's not cellulite, okay, cellulose, knows ose is a sugar, we're still in carbohydrates, and glycogen. Now it's where you find them. So starch and cellulose are found in plants. Okay, make sure you make a note of that, okay, because I may ask you a question dealing with these and mix it up and say, well, cellulose is found in animals. Well, that's not true. They're found in plants. So cellulose makes up the structure, so like the cell wall of a plant. Starch, we eat starchy foods, so that would be like a potato or carrot storing the food in their cells. And then we eat it and then convert that to an energy that we can use. Glycogen is animal starch. So we take that food that we eat, our liver and muscles will then store that carbohydrate so we can use it later for energy. Okay, so just keep those straight. I'm not going to ask you ask you questions about diagrams, but I just want to show you they're still glucose molecules, so each one of these hexose is a glucose, but it's how they're arranged. They're a little bit different in the number and the length. Okay? Okay. The second is our lipids. So that's our fats and our oils, and the building blocks are fatty acid or fatty acids. It changes depending on the lipid, and a glycerol. Anytime you see OL, that's indicating it's some type of alcohol. Okay, so this one's a fatty acid and this one's the alcohol. I'm not going to ask you to be able to identify the structures. Don't worry about that. Just know the building blocks. A lipid has 2 primary functions. One is going to store energy, and the other, which is really important, is to help build the structural parts of a cell membrane. If the cell membrane, which your body is made up of millions of cells, if those start to break down, then you're going to have some major problems, so to speak. Okay, and lipids, I'm sure you've heard this, that oil and water do not mix. Well, it's because the water cannot dissolve, the oil cannot dissolve in water because it's non-polar. Okay, there's no poles involved. The electrons are not shared. The electrons are shared equally, but there's no attraction to help break that apart. So that's why they can't dissolve. Okay, one lipid is a triglyceride. This one has 3 fatty acids and 1 glycerol. Triglycerides are the fats. It's going to circulate in your blood. It's formed by the liver, so anytime you eat something that has a lot of fat in it, then your body's either going to use it or store it. We do need it for our tissues, okay, because it helps to build our tissues. But like anything, if you have too much of a good thing, then it could lead to problems, and in this case, diabetes or heart disease. It starts to accumulate and causing distress. Okay, now there are 2 types of fatty acids. There are saturated and unsaturated. And so depending if it's at room temperature, a solid or a liquid, so a saturated fat would be like a stick of butter, okay, at room temperature. And you find that in your meat and your fish. I'll come back to the bonds in just a second. The unsaturated fats would be a liquid at room temperature, any of your oils. Now the bonds dictates, if it's a single bond, it's more rigid. That's why butter is solid. And if it's a double bond, and down here I have the bonds, if it's a double bond, it's more fluid. Okay, there's more space between it, it can move. So just know the difference between saturated and unsaturated. Okay, now why do we need to have lipids to our cells? The first one is the membrane, the phospholipids. It's made up of 2 fatty acids and a phosphate. So if I come down here, here's the polar head, here's the nonpolar tail. Okay, so this is one phospholipid. When I put them end to end or tail to tail, okay, this structure here makes up the outer of the cell membrane, and you find that in both plants and animals. So that's why lipids are very important, is because of this phospholipid. Okay, another important type of lipid is cholesterol, and that's a type of fat that we find in animals. It's produced in our bodies in the bloodstream. And so we'll look at something called lipoproteins. And so cholesterol is carried in the blood by these special proteins. And cholesterol is really important, especially when you're going through puberty, because it's the development of the sex hormones. Okay, so it helps you to start and continue through puberty. So here is HDL and LDL. You may have heard that if parents or grandparents, cholesterol, triglycerides, if they have a lipid panel. So HDL, HD is high density, the L stands for lipoprotein. So the HDL are your good cholesterol. So what it does not deposit in the blood. Your LDL is your low density lipoprotein, or your bad cholesterol. I had someone call it, and I thought this was real clever, lousy cholesterol. That's a good way to remember it. So this will, if you have a high LDL, it's going to start accumulating in the blood vessels. So if you look at the diagram, this yellow arrow, okay, you see all the yellow starting to deposit in this artery. When it does that, it's building up plaque, which restricts the flow of blood through that vessel, and then that can lead to cardiovascular disease. Okay, so our third macromolecule is a protein. Proteins help with the cells in order to repair itself. It communicates between cells. I know you've heard of talking about hair products and skin and hair, part of our skeleton, enzymes associated with DNA. So protein is very important. We're actually going to have a whole unit next semester dealing just with this protein. The building blocks of protein are amino acids. So it's going to be made up of the elements carbon, hydrogen, oxygen, nitrogen, and some will even contain sulfur. So you will need to know this basic structure. Now it may not be the exact same color code here of the blues and the yellows, but you need to be able to recognize the amine or the amino group, which is a nitrogen and two hydrogens, and the carboxyl group, which is carbon, oxygen, and then a hydroxide. And sometimes they'll write it -COOH. And then you notice they have the carbon chain, the backbone. This one just has two carbons. The variable, this will change, and this is what determines the different amino acids. But everything else will be the same, no matter what kind of amino acid it is. Okay, the variable represented by the letter R, there are 20 known amino acids to date. Now that doesn't mean in the future they won't discover more, but as of now there are 20. And you can see the structures are the same. The variable will change. This one has a particular another hydrogen off of this carboxyl group. And what holds the amino acids together are called peptide bonds. Anytime you see any term related to peptide, you're talking about something related to a protein. So just kind of keep that in mind, especially if you can't figure out a term. A polypeptide would be a long chain of amino acids. So that's what this structure up here, the different kind of balls. When it starts to twist and kind of fold on itself, depending on how much it folds, then that makes the protein. So when it's a long chain held together by peptide bonds, that's a polypeptide chain. Once it folds into the different structures, then it becomes a protein. And we're not going to get into the different, this tertiary structure, so there's different types named by how they twist on them. Okay, the fourth macromolecule is nucleic acids, and that is your DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So it's the amino acid sequence that's going to determine basically the nucleic acids. So it's the amino acids and the proteins, they all work together to make a nucleic acid. Next semester we'll get into how this process, how it actually builds a nucleic acid. But for right now, just know that it's the DNA and RNA. It controls the genetics, basically passed on generation to generation. And the main thing I want you to focus on are the building blocks and what makes up a nucleotide. And then we can reference that later. So nucleotides make up nucleic acid. So nucleotides are the building blocks of a nucleic acid, or nucleotides are the building blocks of DNA and RNA. There are three parts or components that make up a nucleotide. The first one is a sugar. Notice the OSE on the end, that indicates a sugar. Pentose. Pent means 5, so it is a 5-carbon sugar, just like our hexose earlier. Now, depending on the molecule, RNA or DNA, they have their own sugar. So deoxyribose is for DNA. Remember, deoxyribonucleic acid, and ribose is found in RNA. The second part that makes up a nucleotide is a phosphate group. Now this one's found in both. There's no variation. It's not broken down. It's just a phosphate group. So I will ask you questions on a particular part or component of a nucleotide and which molecule it's found in. So make sure you take a look at that. And the third part of a nucleotide is a nitrogen base. Now this seems like a lot, but if you break it down, I've got another diagram that'll kind of put it into perspective. So I've got these 5 bases, and those are the ones that are going to attach to one another to make a DNA molecule. And these bases is like the code for all the genetics and making a protein and everything. Okay, it's a sequence. So we have adenine, which is a capital A, that's found in both RNA and DNA. Thymine is only in DNA, guanine and cytosine is in both, and uracil is only in RNA. So this code, and figuring out what codes for a hair color, eye color, anything in diseases, that's kind of the genome project, and so that's what scientists are working on, is figuring out this code of these nitrogen bases. And what holds these nucleotides together are the hydrogen bonds. There's that hydrogen bonds again, okay? Now here's the chart I was referring to, and it compares and contrasts the DNA and the RNA. So first we have the sugar. We have the deoxyribose for the DNA and the ribose for the RNA. Notice the phosphates are the same. The base is going to vary, so every nucleotide is a little bit different. Notice the color here. Here are the bases right down here. So cytosine and guanine are found in both. Adenine are found in both. But the uracil and thymine are different. Now you notice the shape. Okay, only adenine can match up with thymine in DNA, and only adenine can match up with uracil in RNA. If I start mismatching like guanine and thymine and it doesn't correct that, it can start developing or lead to mutations. Some they're not fatal, but some could be fatal. In between it may be a little hard to see, but like here's thymine and adenine, they're little dots. Those are my hydrogen bonds. And the other thing I want to point out is that you'll notice that here's my phosphate and my sugar, phosphate, sugar. So these are alternating phosphate sugar, and they call that the backbone of a DNA molecule. It's also in the RNA. You notice it's flipped, so I've got my sugar and phosphate. Okay, here are my bases that are connected together. So 2 strands for DNA, 1 strand for RNA. Okay, now we can't talk about the DNA molecule without talking about 4 different scientists. The first 2, Watson and Crick, they proposed the model. They actually built a model of the DNA molecule. It's about 1953. I'm not asking years, just give you a frame of reference when this took place. It's a double helix or a twisted ladder. So here's Watson and Crick. Here's their first model they built, and from that, this is the current model that we use now, that we can actually clip together little plastic balls if you want to. Now, if it wasn't for the work of Wilkins and Franklin, here's Wilkins on the right. Rosalind Franklin was a pioneer in her time. Very bright woman. She knows she only lived to be 38, but her work primarily, she was able to take what's called X-ray diffraction, and it's literally, she was working with X-rays. At the time they didn't know the harm that it caused, but she actually took a paperclip and a strand of DNA, and then she shot an X-ray through that and then captured it, and here's the image here. And then from that she took mathematical formulas, and she was able to deduce that it formed the X pattern or the double helix. Okay, so from that data she was actually able to come up with this, and then from that Watson and Crick built the DNA model. but the reason she passed away from ovarian cancer is because she was exposed to high levels of radiation for extended periods of time. Okay, so this concludes our lecture. Here's the last slide, kind of wrapping everything up. Here are the macromolecules. Here are the building blocks for each. And again, go ahead and look at the online textbook. Take any notes I did not cover from section 2.3 and add to your spiral. And make sure you look over that well in advance before you take the exam. So I'll talk to you later. Bye-bye.